Hello, this is Chemistry News Selection channel. My name is Greg. Today we're going to speak a little bit about nanotechnology. I know you probably expected a video about the Nobel Prize in Chemistry, which was given for asymmetric organocatalysis. A very cool theme, which I will definitely tell you about probably in a couple of weeks from now. But today I have something else prepared about a new way to control the length of nano-cylinders. The links to all the publications are going to be in the description below. First of all, this is the new presentation format. I hope you'll like it. We have Walter White here and of course Dmitry Ivanovich Mendeleev. This picture, something like that, you're gonna see many times when you Google the word nanotechnology. While it's of course very nice, in reality this is of course not the case, and it's more about much more simple shapes which scientists now learn how to control and how to operate with. But before I go to specific shapes and specific chemistry, a couple of words about soap. Why soap? Because the topic today is going to be about structures which form in somewhat similar way to which the soap particles work. And what do I mean by that? Soap works in a way that its molecules have water-soluble or hydrophilic head and water-insoluble or oily hydrophobic tail. So when you put soap in water, it forms structures which are called micelles. It can be like those spherical structures, and the way soap works, it just captures the water-insoluble particles. Like if you had some oil on your hands, the soap will capture those oil drops with their hydrophobic tails and will dissolve it in water with the heads. So it's going to look something like that. Based on this principle, scientists tried to make nanoparticles by self-assembly from block-copolymers. What is block-copolymer? Polymer is a long molecule in which you have repeating units. So let's say you have repeating units which are soluble in water, for example like here, like blue, and repeating units which are not soluble in water, like red. You put them in water, they will form structures like soap molecules, but in polymers you can control the length of each of its blocks, right? And there are many other features like the level of polarity, the level of solubility, or functional groups on the surface of the molecules. And by controlling these, you actually can control the structure that you will obtain. Here are examples of those structures. So it can be spheres, like here with the soap. It can be vesicles, so some spheres with empty space inside. It can be ribbons or two-dimensional sheets. Those are the major structures. And out of those structures, this one, the cylinder or worm-like structure, was very hard to obtain. There was basically no ways to really study those structures before those gentlemen came into picture. And this is Professors Ian Manners and Mitchell Winnick. I had the honor to work with both of them. So what Ian discovered around the end of 90s is that if you take a block copolymer in which one block is not just self-assembled in the solution, but crystallizes in the solution, where the molecules interact not just randomly, amorphously, but fold in some sort of order, then you can get selectively cylinders. So in this particular case, I talk about the polymer called PFS, polyferrosenilsilane. This is a pretty cool molecule, which maybe I'll discuss in some other video. And you have the repeating units of this PFS. The number of repeating units is represented by the letter M here. And the other block, the other part of the polymer is not crystallizable, and this is called polyisoprene in this case, or PI. And the self-assembly here happens not in water, but in a non-polar solvent, in which PFS crystallizes, like decane or hexane. You just take a solid polymer, you put solvent on top of it, heat it, it dissolves, then it cools down, the polyferrosenilsilane crystallizes, and you get cylinders. This is a picture from transmission electron microscope. You get very long random length cylinders. So another thing that was discovered, that if you take the solution of those nano-worms and sonicate them, then instead of those long micelles you get those tiny fragments. That's schematically represented here. Oh, this is, by the way, a very nice picture from the microscope. You can even see the core, which is darker because PFS has iron here, so in the transmission electron microscopy it will be darker. And you also have the this block, which is lighter, the polyisoprene. It's swollen with solvent. So those fragments are called seeds. Why they called seeds? Because you can add more of this polymer to the solution and it will assemble on top of those seeds, and by this you will be able to control the length of those cylinders. You may ask now why it adds on top of the seeds. Why it does not form new seeds? Why it does not make, again, random structures like on this image on the left? So the reason for this is that in order to crystallize, first nucleation should occur. That means several polymer molecules should form an initial crystal. This process is long, but when the initial crystal formed, other polymer molecules which are dissolved and did not form the nucleus yet, they will add to this nucleus, and this process is much faster. So if you have already crystalline nuclei existing in the solution, if you add block copolymers, they will add to them. They will not have enough time to form new nuclei. And that's what happens. This way you can not only make structures with the controlled length, but you can also make block co-micelles. What do I mean by block co-micelles? Then you not take necessarily the same polymer, like here PFS, PI, but you can take another block copolymer in which you have again PFS, which will crystallize on to the other PFS, but the second block will be different. In this case it's called PDMS, polydimethyl siloxane. And this way you can form micelles with different regions, chemically different regions. On one region you can do chemistry and the other you can just live without chemistry. And I'll show you some examples why it's important. And here this image of micelles of similar length, which has three blocks. The PFS-PI block in the middle is darker, and PFS-PDMS block on the sides. Well, if it's not convincing, I'll show you something that very convincing, and this is another paper. This paper, by the way, was published in Science in 2007, and this one from Nature Communications in 2014, in which the group of Ian made colorful blocks. So this was in transmission electron microscopy, and this one is a confocal microscopy. So they put dyes which emit light on different wavelengths, and they made those micelles with different blocks of different colors. I will not get into this work too much, but you just look, you can see the different blocks distinct in those microscopical images. Another interesting thing you can see here is that it's actually not symmetrical. So there's a way that you can seal the seed from one side and grow it from the other side selectively. This is a theme for another lecture. This was another paper in Science. Besides just taking seeds and adding another portion of block copolymers, there were other ways to control the size of the cylinders. And this is the work from 2011. Apparently, if you take the solution of seeds and heat them, some of the seeds dissolve, but others remain in solution. And when you cool it down, the dissolved polymer adds on the seeds which survived, and you can get micelles again with uniform size. And you can control the size of those micelles amazingly well. So I put here also heating or THF addition. THF is a solvent that dissolves both the corona block, the PI or the PDMS, and the core block, that means PFS. It dissolves the whole seed, and if you add a little bit of THF, it will also dissolve part of the seeds and not dissolve the others if it's a very small amount of THF. And then you can selectively remove THF because its boiling point is lower than, for example, decane, and THF will be removed, and you again will form uniform micelles. Here you can see a very nice chart of length as a function of temperature to which you heat the seed solution. Very similar chart you can build of length as a function of THF concentration which you add. These are fragments or seeds. This is cooled from 70 degrees and these are cooled from 80 degrees, much longer because more seeds dissolved. Also here you can see the distribution of lengths, schematically described by those additional lines around the dot. So you can see the precision of the length control that can be achieved. Another way to control length, which actually was discovered in my work in the group of Mitch Winnick, was the control by concentration. So if you take a different concentration of the seeds, you will get different lengths. Here is the chart of the different lengths as a function of concentration of the seeds. And to speak as simply as possible, when you have more seeds per volume and they kind of interfere with each other, so it's more difficult for the solvent to dissolve the seeds, so more of seeds survive, and if more seeds survive at higher concentration, you will get shorter micelles. Just to summarize what I told you till now before we go to actual new publication from Ian's and Mitch's group. So there's several size control methods I showed. So first, in all of them, you have first to dissolve in the block-copolymer, get the micelles of different lengths. Then you have to sonicate, get the seeds. Then you can either add more block-copolymer to the solution to get those micelles, or we can do the self-seeding, control either by temperature or by solvent, HF addition, or by concentration. The question is here, can we do it by a single step? Just take the polymer and heat it, dissolve it, and get narrow length distribution. Seems like a hard task, but this exactly what was done in the recent publication in Journal of American Chemical Society, also known as JACS. Before I show what was done, maybe you want to try to think. Just remember that what I told you from the beginning. Nucleation process is very slow. After you have the nucleus, you will have very fast addition. So the question basically, can we do controlled nucleation, quick formation of limited number of the seeds on which the polymer can add? If you was able to come up with some idea, this is just amazing. And for those who want to enjoy the show, that's what was done. The idea is just to add a little amount of pure polyferrosanil silane. Not the block-copolymer, but homopolymer, which crystallizes very fast. Basically, what prevents from the block-copolymer to crystallize quickly is the corona, right? Is the soluble block. So if we take a PFS without the soluble block, a really small amount, but a known amount, of course, it will form the initial crystals much quicker than the block-copolymer. And that's what was discovered. And by concentration of the PFS that you add to this solution, you can control the final amount of nuclei and the final length of the micelle. Those graphs show exactly this. The length as a function of the temperature with different amounts of PFS added, and this is the length as a function of PFS added. It was also very nicely visualized by, again, this confocal microscopy. They just added the red dye to the homopolymer and the green dye to this block-copolymer, so you can see the red core, which is the crystal of the pure PFS, and the green tails on both sides. Very uniform micelles. Just incredible thing. And not only that, using the same idea, was able to make block co-micelles. I'll show you what I mean. So here we want to make either block co-micelle in which in the middle we have PDMS and the PI we have on the ends of the micelle, or we want to make exactly the opposite. So in the middle we have PFS PI, it's here blue, and on the ends we have PFS PDMS. Can we do it in a single step? The answer is yes, we can. So the idea here is to take block copolymers with different solubility. So after the initial nuclei forms from this homopolymer, the PFS 35 in this case, in both cases, the block copolymer, which is more soluble, will remain in solution, while the block copolymer, which is less soluble, will add on the nuclei first, and the more soluble will be added after that. That was the idea. PFS PI, that have PFS 35, which is quite short, but here we have PFS 90. PFS 90 is already quite insoluble block, so it crystallizes first on the seed, and this way we have first crystallization of PFS PDMS, and then the PFS-PI adds on the ends. Here on the right, the story is opposite. The PFS-PI unit has 74 PFS units, so it's much less soluble than the PFS-26. So the PFS-PI will be added first on the nuclei, and the PFS-PDMS will be added after. And here you can see the microscopy. So here we have a very long PFS-PDMS core of the micelle and the shorter PI. Here we have the opposite way, so we have longer PI and the shorter PDMS, all based on the concentrations of the polymers that we add. And here again, same idea for the opposite structure. We have a very long PFS-PI in the middle and the shorter PFS-PDMS on the ends, and here the PFS-PDMS part is also very long. So amazing level of control, again, all in single step. So that's the chemistry news I wanted to tell you today. If you ask, well, but you didn't tell us where we can use all that, why we need those cylinders at all, how you can sell them. Well, we still cannot sell them, it's still not commercial use, but there's many ideas where it will be possibly used in the future. And also very nice works from Ian's group about how it can be applied in nanomedicine, in catalysis, nanoelectronics, and so on. There's a paper, actually a review paper about these uses that was published also very recently, but it will be a subject for a completely different video. This was already too long, and I personally preferred the fundamental stuff. I mean, applications are always nice, but not as nice as the hardcore fundamental science. So thanks a lot for viewing this video. I hope you liked it. Please subscribe to this channel. I'm trying to post a video once in a couple of weeks or once a month at least about different publications. Please like this video so YouTube algorithms will suggest it to more people. Have an excellent day and see you soon.